Hey everybody, welcome back for part 3 of our nephron transporter chapter. So now we're going to talk about the third part of the nephron, which is the distal convoluted tubule. So as you remember, first we talked about stuff is filtered at the glomerulus, then it goes to the PCT, then the loop of Henle, and now we're at the distal convoluted tubule. So here's our image again. Remember the first video we highlighted the PCT in our yellow highlighter. In the last video we did the loop of Henle in our green highlighter. And in this video we're going to use a blue highlighter to indicate the portion of the nephron that's approximately the DCT, or the distal convoluted tubule. So right off the bat we're always thinking about what is the function of this portion of the nephron. And it's really got a lot of similarities of the other portions of the nephrons that come before it, in that we're trying to reabsorb solutes, right? So that we have a proper amount of ions and nutrients that we need in our body. And at other parts of the nephron, we're trying to reabsorb that water volume as well. First thing I want you to notice here is that this is impermeable to H2O. That should make you think back to the loop of Henle and say, on the ascending limb, the thick ascending limb, it was also impermeable to H2O while it was actively transporting sodium chloride into the bloodstream, into the peritubular capillaries. And so if you think about that being a continuation, the fluid has now gone up the loop of Henle and the distal convoluted tubule. We're still pulling out solutes. We're still pulling out sodium and chloride, but it's impermeable to water. And that is why this is the area of the lowest osmolarity. I put that in there because I think it's particularly high yield, and I've seen them ask questions about that a lot. Remember, as we go through the nephron, the osmolarity is continuously changing, right? So if you follow my red arrow at the bottom, an average osmolarity is approximately 300 milliosms when it's filtered. This is typically the same around the PCT, but as we get down towards the lower portion of the loop of Henle, it gets really, really concentrated because remember, in the thin descending limb, we are reabsorbing water. So let's say it's around 1,000 milliosms. And then as we go back up the thick ascending limb, we are actively transporting solute out of the nephron in NaCl, so it becomes more and more dilute because we are diluting it and that we are taking out solutes. And so you can see, because the DCT is essentially a continuation of this process, this is where we get the lowest amount of concentration of solutes relative to the amount of water. Because now we've gone through this pretty long portion of the nephron in the thick ascending limb, plus the distal convoluted tubule, which both are reabsorbing sodium chloride, but are impermeable to water. And thus the milliosms around there could be anywhere from 100 to 200, which is pretty dilute. Also notice the similarities in that sodium chloride is reabsorbed, just like in the thick ascending limb of the loop of Henle, but also magnesium and calcium are reabsorbed. Difference here is that in the loop of Henle, we talked about this idea of paracellular transport and how potassium leaks out and creates an electrochemical charge that pushes it across in between the cells. But here in the distal convoluted tubule, we actually have some different transporters that allow these molecules to move within the cell. So I'm going to highlight these in pink here to really draw attention to them. So you can see that these are transporters that allow these large positively charged molecules to actually pass within the cell. These are transporters that we did not find in the loop of Henle, which is why there was a different mechanism for reabsorbing these ions. Another very important and high yield thing for you guys to know about the distal convoluted tubule is the effects of PTH. Recall, PTH is called the parathyroid hormone. We've talked about it in a previous lecture where I called it the phosphate trashing hormone, because one effect of PTH is that it actually causes us to excrete more phosphate in our urine. However, remember that PTH is also the primary signal the body uses to indicate that we need to reabsorb more calcium, or that we are low in calcium levels and we want to take it out of the urine and keep it in the body because we need it for various reasons. So I'll highlight the PTH receptor here in the light blue, and you can see that when this molecule acts, its primary function is to stimulate the calcium reabsorption mechanisms, the calcium transporters, to bring that calcium in the body. It's the signal that says, hey, we're low on calcium. Don't let it get away. Don't let it be excreted in the urine. So on the diagram, you can see I drew a plus sign indicating that the PTH binding to the receptor is stimulating the calcium to be reabsorbed from the nephron, and then ultimately stimulating the calcium sodium transporter to actually bring that calcium back into the body. And the final thing that we're going to talk about in this video, again, another renal tubular defect. So far, we've talked about Fanconi syndrome at the proximal convoluted tubule. We talked about Bartter syndrome in the loop of Henle, and now we're talking about Gittleman syndrome in the distal convoluted tubule. So Gittleman syndrome is a reabsorption defect of sodium chloride. Remember, this is one of the primary jobs of the distal convoluted tubules to bring this back. So if it's not working, if transporters aren't working and the mechanism is impaired, this is a condition that can cause some problems for a patient. This is similar to the action of thiazide diuretics, which we can use to actually inhibit these channels in the distal convoluted tubule. We'll talk much more about that in the pharmacology section. Notice some of these labs and exam findings that we can see, such as a metabolic alkalosis, low calcium, low magnesium, and low potassium levels. And again, this has to do with the fact that if these transporters aren't working in distal convoluted tubule, we aren't able to get these substances back that we would like to have in the body, and thus they are excreted in the urine. So we have low levels because we cannot bring them back without the transporters working properly. And finally, this is also an autosomal recessive condition. So like I said last time, just remember the genetic conditions in the nephron are autosomal recessive, except for Little syndrome, which we'll talk about in the next video on the collecting duct, which is autosomal dominant. So if you just remember that's the one that's different, that's the one that's unique, and the rest are autosomal recessive, it might be a little bit easier to keep those in your memory and answer your questions right on test day. All right, that's all I have for this one. I will see you all in the next video. We're going to finish the chapter on nephron transporters, talking about the collecting duct.